Zinc is an essential trace element and is necessary for proper immune function, gene transcription, and protein synthesis. Zinc is a cofactor for many enzymes and is important for the function of zinc-binding proteins. Zinc is found primarily in the protein and nucleic acid fractions of foods. Intestinal zinc absorption is an essential component of whole-body zinc homeostasis. Within the enterocyte, two families of zinc transporters, as well as the zinc-binding protein metallothionein, work to control zinc movement across the intestinal epithelium. There are two families of zinc transporters. There are 10 SLC30A members. SLC is abbreviation for soluble ligand carrier. These members are also designated ZNT transporters. The ZNT transporters transport zinc out of the cytosol. There are 14 SLC39A members. These members are also designated ZIP transporters. The ZIP transporters transport zinc into the cytosol. Both families of zinc transporters function by facilitated diffusion. The ZNT transporters transport zinc from the cytosol across the plasma membrane to the cell exterior or from the cytosol into intracellular compartments. The ZIP transporters transport zinc across the plasma membrane into the cytosol or from the lumen of the intracellular compartments into the cytosol. Zinc transported into the enterocyte enters the intracellular zinc pool. This pool is made up of zinc-dependent enzymes and zinc-binding proteins. Intracellular zinc can also be transported into and out of intracellular organelles via the zinc transporter family members. Metallothionein is an important zinc-binding protein. It is a low molecular weight, cysteine-rich zinc-binding protein. It is induced by zinc and restricts zinc absorption in conditions of zinc excess. At the basolateral membrane, zinc is transported out of the enterocyte by ZNT1. Zinc then binds to albumin, which is the major zinc carrier of the plasma. As the zinc concentration of the intestinal lumen increases, such as with the consumption of zinc supplements, the ability to transport zinc across the cell from the apical brush border membrane to the basolateral membrane decreases. The contribution of simple diffusion to intestinal zinc absorption increases with increasing luminal zinc concentration. Zinc diffuses in between the cells of the intestinal epithelium as it moves through tight junctions. Once it crosses the tight junctions, it binds to albumin in the plasma. Patients with acrodermatitis enteropathica produce defective ZIP4 transporters, so zinc is not transported into the enterocyte from the intestinal lumen. Patients are then treated with zinc supplementation in order to overcome this defect. Zinc is also transported from the body across the intestinal epithelium into the intestinal lumen. ZIP5 transports zinc across the basolateral membrane into the cell. This zinc enters the intracellular zinc pool or is transported out of the cell at the apical membrane by ZNT5B.